Okay. What I'm going to show to you is the work we performed in the last 5-6 years with our group of the Foundation on Endometriosis. And next slide. Okay, just few slides in order to make you comfortable with this argument, endometriosis. This is the classic definition of this disease. Endometriosis is a gynecological disease defined by the histological presence of endometrial glands and stroma outside the uterine cavity. It can be found in several parts of the body, the surface of the exothelium surface, but also deep inside the body, also in places very far from the uterus. There are also cases in the lung, in the skeletal muscles, in the brain even. C'è? Potete sentire, sì?
Ma un po' di voce, un po' più alto.
Ok. Okay. Which are the numbers of endometriosis? 150 million women affected all over the world. 3 million only in Italy. Annual healthcare costs very high. But, respect to this, the investment in research is very low. This is data from a few years ago in the US. Only 90 cents for each patient. So, which are the consequences of these numbers? If you do a very, very simple research on PubMed, that is the most well-recognized database to look at scientific paper for us, and you digit the word cancer, you find millions of papers. If you digit the word Alzheimer, that is one of the most common neurodegenerative disease, you find several thousands papers. If you digit endometriosis, you find much less papers. These are data. I did this research a few days ago. So less money, less investment, less research. So the consequences of these numbers, which are the pathogenesis is still not clearly defined. We are in 2012 and we have a disease so common where the pathogenesis is not clear. And therefore there is not a definite therapy. Actually, there is not a therapy for this disease. The drugs actually used are not suitable for prolonged treatments and have important side effects. Moreover, the pathophysiologic mechanisms of the associated infertility, that is one of the most important problems for these patients, are not clear. So which are the theories, because we have to talk about theories, on the pathogenesis of endometriosis? The most fashionable one, the one that is still most accepted by the scientific community, is the Sampson theory, made up in 1922, where it's claimed that retrograde menstruation is the cause of endometriosis. Then, at the end of the last century, came up this theory about the coelomic metaplasia that claims that there are some immature cells that in a certain moment start to form endometrium in the body. Then, something like 10 years or 8 years ago, came up the theory of endometrial stem cells, or transient amplifying progenitor cells. This theory supposes that there are some stem cells that are able, in a certain moment in the adult life, to start to grow again and build up the endometrium. And then there is another theory, this is the embryogenetic theory, that was made up coming down two centuries ago. This theory suggests that endometriosis is caused by microscopic remnants of a woman's embryonic endometrial tissue from when she was an embryo that later develops into endometriosis. So, and this is the theory that was set up by the pioneer scientists in medicine, pioneer pathologists like Boehringer-Ingelhausen, like Pick or Cullen or Frank. Then, starting from the observation of Sampson, this theory was discarded and never studied again. Based on the great clinical experience we have with this disease, thanks to the work that the Centro Endometriosi does every day with these patients, we have been convinced that the retrograde menstruation theory is not able to explain all the clinical cases that we have seen. And so we decided to go through deeply on the embryogenetic theory, work that has never been done. So, if the basis of endometriosis is an alteration during organogenesis, it would be possible to see ectopic endometrial tissue dislocated outside the uterine cavity in fetuses. In order to do this, we set up a very time-consuming and hard work. We enrolled at autopsy a total of 101 female fetuses from 20 weeks of gestation to newborns. Then we collect the pelvic organs of these fetuses and block, and then we made analysis by histology and by immunohistochemistry. So a classic morphologic approach. And then what we found, we published this data in several scientific papers in the last 3 years. We found in a total of 9 over 100 cases, so the 9%, ectopic endometrium outside the uterus. The endometrial nature of the tissue was confirmed both by histology and by immunohistochemistry. Expression of estrogen receptor, or CA125, or cytokine 7, and CD10 in the stroma. And then I show you briefly some of the cases. This case is close to the posterior wall of the rectum, the gland here. And then in this case, we see here a little spot of adenomyosis, endometrium in the wall of the uterus, and then some glands in the posterior wall of the uterus. This case 5 here, deep in rectovaginal septum. This is an example of all the immunohistochemical analysis that we did on these samples. This is hematoxin. Then we have the CA125 and the estrogens. Here you have the CD10, the cytokine 7, the CD10 in the stroma. These are the other cases, hematoxin, eosin, and then this is the estrogen and the CD10 staining. And then another observation, the ectopic endometrium displays the same phenotype of the ectopic endometrium. So another proof that this tissue that we are seeing is close and very similar to endometrium. Cytokeratin, estrogen receptor, CD10 in the stroma. This is just a schematic representation of where we found all these different cases of fetal, we call endometriosis. So generally, as you can see, deep endometriosis, generally along the posterior wall of the uterus or in contact with the rectum. So, based on this, we started to speculate why this happens, which is the molecular mechanism involved in this. And we made a simple protocol, which is the most important embryogenetic factor during the early life in a woman, especially for the genital tract, is estrogen. So maybe acting on the estrogen pathway, even at molecular level, could be possible to cause this little alteration in the formation of the uterus, and therefore the endometriosis. And which can be a method in order to investigate this, using endocrine disruptors. I don't know how many of us are familiar with endocrine disruptors. Anyway, this is the classic definition. So they are chemical substances that alter the function, the structure, or maybe better, the functions of the endocrine system. This means that they act like xenoestrogens, like you put estrogen inside. Endocrine disruptors are present in our everyday life. they are the component of all the plastic material that made up the bottle of the water and all the other box where you put the food. So we have a lot of contacts with endocrine disruptors. And among the several endocrine disruptors that are present, we choose BPA, bisphenol A. Why bisphenol A? For two reasons. One, historic, because it's the firstly reported synthetic chemical causing estrogen receptor modulation. And second, because bisphenol A is one of the most common endocrine disruptors present in our everyday life. And it accounts for the majority of the estrogenic activity that leaks from landfills into the surrounding ecosystem. So, this is the background from where we started. I mean, we were not the first to look at the endocrine disruptor, in particular to BPA. There are several studies published in the literature that reported that endocrine disruptors can affect at very low doses the endocrine system and the development of mammalian and non-mammalian species. So we are not talking here about a toxic effect, but effect at the molecular level. And if you go in the literature and look it, you find an increasing number of low-dose studies in mammals that have implicated perinatal BPA exposure in a variety of abnormalities in the female reproductive tract: early onset of vaginal opening, early onset of puberty, altered estrous cyclicity, altered plasma levels of luteinizing hormone, altered vaginal, ovarian, and uterine histology. And also there are papers coming out now that link it to a higher frequency in some cancers, breast cancers, uterine cancers. And also in other apparatus. For example, neocortical development. There are studies that are trying to link exposure to this kind of substances to some pathologies of the brain, like autism. Then, are there some links between endometriosis and endocrine disruptors? Yes. There are several studies in humans linking exposition to endocrine disruptors with insurgence of endometriosis. In particular, this is a study performed some years ago by the group of Dr. Mismer from Boston. It has been found in a wide cohort of patients with endometriosis that the rate of endometriosis is 80% greater among women exposed to the endocrine disruptor in utero. That means that their mother has been exposed to an endocrine disruptor that is diethylstilbestrol. So there is a clear epidemiological link between these two things. So what we did, very simple, very strike. We took mice, BARC, we treated them with BPA at 2 different concentrations on day 1 of gestation through the 7th day after delivery.